All right, so getting right into it, mitosis. Your lecture notes were fascinating, and it's like, I don't know, have you ever seen one of those perfectly synchronized dances?
Oh, yeah.
It's like that, but inside our cells, constantly happening.
That's a really cool way to think about it.
Mm.
It's easy to forget that at the core of it all, it's this process, mitosis. That's how we grow, how we repair, how our bodies even stay alive with,
Uh,
what, trillions of cells.
right, it's mind-blowing. So we're essentially talking about a cell making a copy of itself.
Yeah,
Like
exactly.
hitting artial plus C and cotrol plus V, but like with DNA and on a microscopic level?
And just like you would want your computer to make a perfect digital copy,
Yeah.
right? Our cells have these incredible ways of making sure that the copy of their DNA, those chromosomes, they're flawless, like absolute precision.
So these notes mention the cell cycle. Is that like the schedule of events and mitosis is just like one of the things on the schedule?
Yeah, you got it. So the cell cycle is basically the entire life of the cell from when it's born to when it divides. And mitosis is the main event, where that cell focuses on dividing its nucleus and all of that DNA.
Okay, so mitosis, it's the main attraction. I also see cytokinesis in the notes. What's that about?
Think of it this way. If mitosis is the main act of the show, cytokinesis is the grand finale, when the cell actually splits into two new daughter cells.
I like it. Okay, so we have our stage set, but before the show starts, before the curtain rises, we got to meet our dancers. The notes mention chromosomes, microtubules, and motor proteins. A lot of big words, but they're like the key players, right?
Yeah, so picture this. Chromosomes. They are the stars of the show. They're carrying all that DNA all bunched up. And to make it even easier, they condense during this process into these compact shapes, almost like, I don't know, sausages.
Easier to move around on stage like that, right? What about the stage itself? That's going to be the microtubules.
You got it. Microtubules. They actually create this dynamic sort of scaffolding. We call it the mitotic spindle.
Hmm.
And that is where our chromosomes do their dance. This spindle can actually change throughout the process. It grows and shrinks as needed.
So it's like controlled chaos on a microscopic level. And what about the choreographers? Who's making sure everyone's like hitting their marks? That's got to be the motor proteins.
Exactly. Motor proteins, they're like tiny little machines that move the chromosomes around using the microtubules as their tracks. They keep the whole process in sync, make sure it all goes according to plan.
We've got the dancers, the stage, the choreographers. Time for the show. The notes lay out 5 main stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. Wow, where do we even begin?
Well, it all starts with prophase. Our chromosome dancers are getting ready for their big performance. They condense and become visible, like we can actually see them under the microscope. And at the same time, the microtubules, our stage crew, they're busy building the mitotic spindle.
Like building a set for a big Broadway show. I can picture it. And there's got to be a director involved somewhere. The notes mention centrosomes. Are those our directors?
Good eye, you're spot on. Centrosomes, they're like the stage managers, actually. They duplicate early on and move to opposite ends of the cell. So, taking positions at the edge of the stage, they're going to be calling the shots, making sure everything's set up right for the main performance.
Prophase is all about setting the stage, getting ready, what comes next in our intricate cellular dance.
So that's where prometaphase comes in. The curtain's rising, things are about to get interesting. Now, do you remember those protein structures on the chromosomes, the kinetochores?
Yeah, I think so.
Well, get ready because they're about to take center stage,
Yeah.
literally. In prometaphase, specialized microtubules called kinetochore microtubules, they attach to those kinetochores.
So it's like each chromosome is being like picked up by these tiny little hands.
Exactly. No more just floating around. Now they're being guided into place.
Tied where exactly?
Think of a line forming right down the center of the stage. That's what we're aiming for.
So prometaphase is lining everyone up for a big finale. What happens when all the chromosomes are in position?
That, my friend, is metaphase. All those chromosomes, all lined up perfectly at the center of the cell. It's called the metaphase plate, and I gotta tell you, it's a really cool thing to see, even under a microscope.
So organized, like a line of dancers all ready to go. And they're not just lined up randomly, are they?
You're catching on. Remember how each chromosome is basically a copy, like a duplicate. We call them sister chromatids. And get this, they're still attached at this point.
So it's like all our dancers are holding hands with their twin, just waiting for that signal to let go.
A very precisely timed signal. That happens during anaphase, probably the most dramatic part of the whole show. Imagine like a drum roll as the spotlight hits the center stage, because this is where those sister chromatids separate.
Okay, but how? How do they actually separate? That seems like a really delicate process.
Oh, it definitely is. So there are these tiny little proteins, cohesins, they're like molecular glue, holding those sister chromatids together. Then, in anaphase, an enzyme called separase comes in, and this is cool. It basically snips those cohesins apart, like with scissors.
Cutting the rope in a tug of war, and suddenly each side is free.
Precisely. The sister chromatids are pulled apart by those microtubule ropes we talked about, and they each head toward opposite poles of the cell. Remember our stage managers? They're making sure each side gets a full set of chromosomes.
This is amazing. And we don't even realize it's happening, but constantly, it really makes you think about how complex everything is, even just inside one cell.
Right. And we're not done yet. We still have telophase to go, where everything gets all tidied up. Remember how the nuclear envelope broke down way back in prophase? Well, it makes a comeback in telophase, kind of like a curtain closing on the act.
The cell rebuilds the whole thing around each set of chromosomes.
Yep. Now we've got two new nuclei, each with all the genetic instructions they need. Oh, and those microtubules. They're disassembled and, like, recycled. Very efficient.
So the cell is ready for the next performance. What of the cell itself, though? I mean, it's still just one cell. That's where cytokinesis comes in, right? The big finale.
Exactly. Think of it like a magician perfectly slicing a lemon in half. Poof. Two identical halves.
Okay, I can picture that. But instead of magic, it's actually a bunch of different proteins working together, right?
The notes mention something about a contractile ring, and actin and myosin.
You're on a roll. So yeah, actin and myosin, those proteins, they work together to form this ring that tightens right around the middle of the cell. Picture a drawstring bag slowly closing.
So it pinches the cell in half until we have two separate cells, each with their own set of chromosomes, ready to start the cycle all over again.
And they do, over and over again. And all of this is like perfectly orchestrated by those molecular machines we talked about. Motor proteins, you had some questions about kinesins and dyne, right? Yeah, I'm really curious about them now. So they work together. It's like a team effort to move everything.
They're like the stage hands. Remember we were talking about microtubules as the stage. Well, kinesins and dynin, they use those microtubules as tracks to move stuff around, like tiny little motors.
Like little trucks moving around backstage. What exactly are they carrying?
Well, a lot of things. But one of the most important is those chromosomes. So, for example, we have Kinsin 5. Kinsin 5 is a real workhorse. It helps to push the spindle poles apart. Imagine, like, two people on opposite sides of one of those collapsible telescopes pushing outward.
The poles are like the ends of the telescope and the Kinsin doing the pushing. What about the other Kinsins? The notes mention Kinsin 4 and Kinsin 10.
Oh, yeah, they're important. Those are sometimes called chromokinsins, and their job is to carefully move those chromosomes toward that metaphase plate we talked about.
Yeah.
Picture like one of those little motorized carts that pull people around an amusement park.
Okay, that's a good visual. And then there's Kinsin 13. What does that one do? It seems to have a totally different job.
It does. It's like the demolition crew. Kinesin 13 helps to break down microtubules, which sounds bad, but it's really important to make sure the spindle is the right length, that the chromosomes move precisely during anaphase.
So many moving pieces. It's amazing that it ever works correctly. We've got all these different kinesins, and then there's dyning. What's its role in all of this? Dynin. So kinesin is like the one pulling everything in the right direction, but dynin, it does the opposite.
Yeah, you could say that. It provides a counterbalance. Imagine like a tug of war happening on a microscopic level. You need both sides pulling to make it work, right?
Definitely. Balance. But where exactly is dynin doing all this pulling from? The notes mention something called the cell cortex, whatever that is.
Good catch. That's its anchor point.
Yeah.
Imagine the cell cortex as the inner lining of the cell membrane. That's where dynin often sets up shop, pulling on those spindle poles, making sure they stay in line.
This whole process, honestly, it's like watching a perfectly choreographed dance. It's kind of hard to believe it's happening inside of us all the time, and we have no idea.
Right. And usually, mitosis is incredibly accurate, but what happens when it's not? What if one of our dancers messes up? If a chromosome ends up in the wrong place?
I imagine that would be bad, like really bad for the cell.
Yeah, definitely not good. That's when you can end up with cells that have either too many or too few chromosomes. We call that aneuploidy.
And that's what causes...
What can lead to a lot of different things, sometimes developmental problems, and in some cases, even cancer.
Oh, wow. So cancer can be linked back to problems during mitosis.
It can. You see, with cancer cells, their cell cycle is all out of whack. It's like the choreography has gone out the window. The dancers were just replicating like crazy with no sense of order.
So those motor proteins we talked about, they're pretty important for making sure things don't go haywire.
Essential. And, the more we learn about all of this, about how mitosis works, the better we might be able to fix those mistakes. Maybe even prevent them from happening in the first place.
It's like we're learning the dance so we can teach it to others, maybe even help them if they miss a step.
Exactly. It all comes down to understanding the role of each of those players, the chromosomes, the microtubules, the motor proteins. It's like learning a new language.
The language of life, well, that's quite a journey. Today, we dove headfirst into the amazing world of mitosis. We talked about those incredible steps of cell division, all those key players, how it all comes together to create two identical cells from one.
We saw those chromosomes condense, the spindle apparatus practically build itself up and then disappear, and those motor proteins like tiny little construction workers directing all the traffic.
It's honestly mind-blowing. Thanks for joining us on this deep dive, everyone. Until next time, stay curious.